All right, good afternoon, everyone, and welcome to today's NAVBO webinar sponsored by the Education Committee. I'm Dr. Amber Stratman from Washington University in St. Louis, and I will be moderating today's session. So I'm pleased to welcome our speakers today, Dr. Gwendalyn Randolph from WashU as well, and Dr. Amreen Mughal from the University of Vermont. Before we begin, I'd like to remind you that all NAVBO activities are now easily found in the calendar events on our website, and please be sure to register for some of these upcoming events. Throughout the presentation, please type your questions in the question box. These questions will be answered at the end of each talk. Our first talk given by Dr. Randolph will be recorded and archived on the NAVBO website for future viewing. However, the second talk by Dr. Mughal will not be recorded today. So our first speaker today is Dr. Gwendalyn Randolph. She will be presenting her talk from IVBM entitled Ileitis-induced tertiary lymphoid tissue that obstructs lymphatic outflow from the intestines. Dr. Randolph obtained her bachelor's degree in biology from Temple University and a PhD in molecular and cell pathology from State University of New York at Stony Brook, working with Martha Fury. She then carried out her postdoctoral studies at Rockefeller University and Weill Medical School of Cornell University under the mentorship of Bill Mueller and Ralph Steinman. She spent 11 years on the faculty at Mount Sinai School of Medicine in New York before relocating to Washington University in 2011, where she is currently the Emil R. Unanue Professor of Immunobiology in the Department of Pathology and Immunology. Her laboratory considers how transient cells, i.e. monocytes or dendritic cells, and molecules like lipoproteins out of tissues influence the inflammatory microenvironment. Her work considering cellular and molecular clearance through lymphatics has fostered key contributions to the understanding of the pathogenesis of atherosclerosis, with recent efforts in her lab shifting to consider inflammatory bowel disease and perinatal immunity. So, peritoneal immunity, there we go. So welcome, Dr. Randolph, and we look forward to your talk. I'll let you take it away.
Thank you very much. And can you see my screen? Hopefully, yes. Okay,
yes.
So I'm going to.
Yes, we can. Yep, it looks great.
Yeah, so as Amber just indicated, I'm going to focus on lymphatic outflow from the intestine and think about a chronic inflammatory disease in the context of lymphatic biology. So let me just begin by making sure that we're all on the same page with respect to the three major components of the lymphatic vasculature that one would find in mammals. So lymphatics are blind-ended vessels, in contrast to the blood circulation, which, makes a true circle in most organs. And these blind-ended vessels are called initial lymphatics, and they have these discontinuous or button-like junctions that allow for uptake of fluid, leukocytes, macromolecules, often in a receptor-independent manner. And then one will see that these vessels converge into larger vessels, which are called collecting lymphatics, and that's actually what you see here pumping. So depending on the body site and the animal, this pumping will be a way in which lymph can be propelled toward the next structure in the lymphatic vasculature, which is the lymph node itself, and a key site for induction of immune responses. And that propulsion will be mediated in part by muscle cells that are in the outside, shown in green. And a key feature of these collecting lymphatic vessels is the valve, which drives unidirectional flow. So the lymph node itself serves as both a collector of antigens for inducing immune responses in cells like T cells and B cells that are present in the lymph node, but also a filter, and cells and molecules that will leave the lymph node will do so by the efferent lymphatics, where lymph can re-enter the bloodstream at the thoracic duct. So in that light, thinking about the convergence of vascular biology and immunology, you can again see a cartoon of these blind-ended vessels in, sort of a drawing of the whole body in a very much of a cartoon form. But again, I want you to appreciate that these lymph nodes are in a position to potentially protect the blood vasculature by filtering out components that would be adverse for blood physiology. So we know, of course, that septic shock, disseminated intravascular coagulation are all events that are to be avoided, for survival in the bloodstream. One of the key roles of the lymph node that I want to highlight here is that some of the components that might be leaving a tissue could be inducing these unwanted outcomes. So for example, antigen presenting cells might express tissue factor, also called factor 3, which you would want not to be, moving into the bloodstream to promote coagulation. And indeed, the lymph node is a place where antigen presenting cells actually will enter by the afferent lymph, but not leave the lymph node. Furthermore, bacteria and in fact, even emerging evidence that tumors can be captured by the lymph node and prevent dissemination from that point. So this is really an important role in protection of the blood vasculature and convergence with host events. Now, lymph node development has been under study for decades, but the role for lymphatic endothelial cells is just now emerging with the work of persons like Burkhard Ludovic and Tanya Petrova. And a key point that I want to draw here is that lymph nodes often develop, so they're called secondary lymphoid organs, because really the site of immune cell development is the primary organ. The secondary organ is the home in which they carry out a lot of their work, and lymph nodes are one of those. And so lymph nodes tend to develop at junctions of blood vessels during early life or even prenatal life, and key activities are either endothelial cells, so-called lymphoid tissue organizers, or mesenchymal lymphoid tissue organizers interacting with specialized immune cells called lymphoid tissue inducer cells. And one of these key interactions is this pathway here for lymphotoxin beta receptor interacting with lymphotoxin ligands that are expressed on the immune cell, and that leads to an exchange of mediators like production of chemokines, et cetera, that will actually assemble the lymphoid tissue. Now, I'm going to be talking today about tissue that's called tertiary lymphoid organs. And so tertiary would be not a structure that forms in homeostasis or during development, but rather in response to a stimulus, and particularly a chronic stimulus. So in many ways, however, tertiary lymphoid organs are structurally organized with immune cells and specialized vessels that is somewhat reminiscent of a lymph node itself. And of course, chronic stimulation in a lymph node can also cause the lymph node to remodel. But I want to focus here on this tertiary lymphoid type of tissue and just briefly say that we know a fair amount about this in part from the work of Nancy Ruddle at Yale, who spent her career working in this space. And so these are just some very quick slides from some of her work where she notes that autoimmunity, which would be a chronic disimbalance of immune regulation, are associated with a development of tertiary lymphoid structures. And so I've listed a few of those, or you see here in one of her reviews, a number of different autoimmune scenarios where tertiary lymphoid structures containing all of the expected accumulations of immune cells and specialized vessels occur. In addition to autoimmunity, infections that are persistent are also drivers of tertiary lymphoid tissue formation, or they can also form after transplant graft rejection. And again, a key feature here is a persistent inflammatory stimulus. And so what we know from manipulation of mouse gene expression is if, for example, you put any of these stimuli that will call in immune cells or the lymphotoxin alpha beta ligand, which is a ligand for the lymphotoxin beta receptor, under the control of a constitutive promoter, that is sufficient to drive tertiary lymphoid tissue formation, really showing that that these signals, when they're persistent, can be drivers. So we understand a bit about their connection to chronic inflammatory diseases, but there's a lot that we don't know about what they really do in a chronic inflammatory state. So that's what I'm going to focus on today, and I'm going to focus particularly on ileitis. So that's inflammation of the ileum, which is the terminal part of the small intestine. And in humans, ileitis is most generally referred to as Crohn's disease. In mice, we tend to use the word ileitis because we don't want to say that the mouse is exactly the same as the human Crohn's disease scenario. But Crohn's disease in humans is one of the two major forms of inflammatory bowel disease, and you can sort of see an important schematic here. This is the terminal part, again, the ileum. Before the small intestine converges with the large intestine, which is also depicted here. So Crohn's can affect both the small or the large intestine, and the disease affected areas are schematized here as this really bright red spaces. And I think what you can see in the bright red schema is you see actually a narrowing of the intestine. So that's a stricturing. And that narrowing is associated with fibrosis and actually can be a cause of a patient having to have a bowel resection because the narrowing that potentially could lead to bowel obstruction. And so I want to point here to, a more elegant diagram of the intestine, so that these intestinal layers, and in Crohn's disease, all layers could be subjected to inflammatory disease. This would be the area that would be stricturing, but just off of that, so thinking back to the lymphatics and the lymph nodes that would drain this space are going to be out here in this adipose tissue, which is called the mesentery. So that mesentery will house the blood vessels that go in and out of the gut, as well as the lymphatics that come out and the nerves that are relevant to controlling bowel function. And we've known for a while that, in fact, maybe lymphatics are critical in the disease. And so if you go all the way back to when Crohn's disease was first described decades ago, pathologists were noting that lymphangitis, or inflammation of the lymphatic system, seemed to be associated with the disease, and they even used comparisons to elephantiasis of the intestinal wall. But we didn't understand it. However, now I want to just show you histology of what would be the fat. So imagine this is the cross section, that's that mesenteric fat that's coming off of the intestine in an affected region. And we've known that those can be inflamed. So the inflammatory foci are shown here by the arrow. You can see that they're circular in nature or follicular in nature, and they sit next to blood vessels. So here you would have an artery vein pair. And it's going to turn out, as you hear my talk, that little lumen that you see that's associated with the follicle is actually the critical lymphatic vessel that's running toward a lymph node. So again, I want to point to stricturing as a problem in the disease. This is actually a person who's having a bowel resection, and this crinkled area here is actually becoming strictured. So the lumen is going to be narrowed, and that's an area where the fat, which can, again, holds the lymph nodes, and it's where you see this inflammation, is starting to kind of move over, the intestinal wall. So it's fat expansion is associated with the stricturing. And a colleague who really got us into the field, Jean-Frédéric Colombel from Mount Sinai, actually had a protocol where in patients, just before they would resect tissue, they would inject dye. The dye outflow will occur through lymphatics. And normally you would see that go straight down to the lymph nodes. But in patients that have this fat expansion and a stricturing of the lumen, you see that a kind of a strange deviation of the lymphatic outflow, and oftentimes it's associated with the fat. So we got really curious about what's going on with lymphatics in Crohn's fat, again, considering the background and the suspicion that something was going on, but really we, didn't know a lot until recently. And there's been a huge amount of growth in the lymphatic field, and lots of tools now are available. So where we started was in patients who had resections of the ileum because of the complication of potential stricturing. What we did was we actually took slices of the intestine. So here's an example of a slice where we left the intestinal wall on it, and this clear area is fat. So we used clearing techniques of 2-millimeter cut sections on a vibratome to kind of basically see through the fat as it was coming off the gut, where the lymph nodes and lymphatics would be, the collecting lymphatics in particular. And so what we found here, you can see this one specimen with autofluorescence on or off, and here's another specimen where we've actually trimmed away the gut, but it would have been here. And I just want to orient you, this is an actual lymph node that would have drained this region of the gut. And this, shadowy line turns out to be the lymphatic that feeds to that lymph node from the gut. And what captured our real curiosity, that turns out to be those little foci or follicles, were these knobs or pearls that were on this lymphatic. And it turns out that, they're pictured here. This is the lymphatic that's feeding down to a draining lymph node, and the lymph nodes have B cells in red and mostly full of T cells. But these structures that were smaller and really didn't have the side vasculature coming in the section were actually these tertiary lymphoid structures, which are full of B cells, as is characteristic of tertiary lymphoid tissue. And you can see here in this one human specimen, if you just blow it up right here, you can actually see that the vessel is running through it. And if I just briefly go back to this, notice how one side of the vessel is almost always on the side, and then you have this kind of a growth that is almost like making a C shape around the edge. And so now I just want to briefly point out again what I put in the introduction, that many types of immune cells, like antigen-presenting dendritic cells, will migrate to a lymph node, but the lymph node captures them there, does not allow them to disseminate. We think this is in part to protect the vasculature. And so you start to ask questions, well, maybe we know that the lymph node should communicate with upstream tissue to keep even actually anti-inflammatory responses properly programmed. So we started to wonder, if you have these lymphoid structures that are now sitting apparently on the lymphatic, maybe they would capture cells that should be going to the lymph node but now cannot disseminate. So we basically hypothesized that these structures would be obstructive. And we also wondered if the lymphatics that were affected by these tertiary lymphoid structures might actually also leak contents like fat, which would drive this fat expansion. So were they leaky? But really, to address that, we couldn't really make a lot of additional progress in humans after making those initial observations. So we asked if we could study this in mice. And we turned to mouse models of ileitis. So that's, now I'll start using the word ileitis, a Crohn's-like disease in mice. And one particularly wonderful model for ileitis in the mouse is this mouse that overexpresses the cytokine, often associated with chronic inflammation, tumor necrosis factor, and the removal, or so the delta of AU-rich response element in the mouse, in the RNA for TNF, actually allows that TNF to continue to be translated into protein, and so you have an overproduction of TNF when a cell gets stimulated by some kind of chronic trigger. So it turns out that the mouse then develops ileitis, and this was known before we went into this. But what no one had really looked at is the mesentery. So what I'm showing you is a mouse that we've crossed to the PROX1 reporter that would label lymphatics and in red, and you're just looking at a stereoscope image. So there's so many lymphatics associated with the intestinal wall that it's just super bright red. And the downstream lymph node is here. And so this is now a display of the mouse mesentery, the outflow of the intestine. Those outflow collecting lymphatics in the control littermate are seen here. They've got surrounding adipose tissue as expected. And then in the mouse that gets the ileitis or the Crohn's-like disease, you actually can see these little lymphoid follicles like the human, we think, and they're positioned right along the lymphatics, as we had seen in human, and very rich in B cells, again, characteristic of tertiary lymphoid structures. So if we look at them in 3D, we see lots of B cells, lots of macrophages expressing a CSF1 receptor, and live 1 positive lymphatics kind of enveloping the outside. And we stain for muscle here because the collecting vessels have muscle. And so what you see, very much like the human, which I had shown you earlier, these structures are clamped on to the outside of a collecting vessel. And so let's just zoom out a little and look at the lymphatics. You're looking at the mouse mesentery from above. You don't see the intestine. But what you're seeing are the collecting lymphatics. The muscle is in red, and the PROX1 expression is in white, and it's very concentrated typically at valves. So these valves are, pointing in this direction. So flow is probably moving in this direction, and they look kind of, at this resolution, relatively normal. But if you look at where immune cells are accumulating, you can see that immune cells are already accumulating around these valves. And then these other regions where you would think a valve would be, now you see some very distorted PROX1. And that turns out to be lymphangiogenesis, new valves, new lymphatics that are developing at that former valve site. And if we focus on studying the expression of a transcription factor that's really highly expressed at valves called FOXC2, you can actually see that some of these, this is an example of a mouse that has the ileitis and has a tertiary lymphoid structure, that sometimes the valve just stretches across the entire structure. So really, very likely not functional as a normal valve. And so as we began to look at this, closer and closer, in fact, we came to the conclusion that lymphangiogenesis that supports these TLOs to form and kind of starts to envelop them actually starts at a valve, probably because the valves become inflamed, and then the lymphangiogenesis starts. And that's essentially why these structures form right along the collecting vessel, which is, until this point, really the field had not known that tertiary lymphoid structures might be associated with outflow lymphatics. So we were very curious to determine if that might mean that they're really obstructive. And so we did two things to test this. And one was to cross this mouse that gets the Crohn's-like ileitis with a mouse that expresses in every single cell this protein called KIK-GR, or KIK-GR, GR standing for green to red. So every cell is green, fluorescent. And this KIK-GR protein, if it's photoconverted by a laser like a 405 nanometer laser, it will turn from green to red. So this is an area of the small intestine in one of these mice that we had converted to red. And what that allowed us to do is ask, well, immune cells that are sitting here would convert to red, and we could actually ask by using flow cytometry to find the red cells, where did they go? And so if things were operating normally, immune cells would move out of the gut into the mesentery, which you can't see here because we haven't crossed it to a molecule that you could see very easily. Then they would move to the draining lymph nodes and again out to blood if they were immune cells that could leave a lymph node, and that could allow them to move to places like the spleen. And we know for lymphocytes that, in fact, some of them may be programmed to return to the tissue and protect the tissue from which they originated from potential additional immune insult. So in the wild type littermate, you essentially see what you expect to see. You see that cells that were stimulated here to turn red, many of them 24 hours later could be found in the mesenteric lymph node, and many more even in the spleen, and some had even returned back toward the intestine. So it turns out, however, that if we looked in the mouse with the tertiary lymphoid structures that developed along the lymphatic, that TNF delta ARE mouse, most of the cells were essentially trapped. So they left the gut and they got trapped in these first location, the mesenteric TLOs, and therefore couldn't really make it down to the other structures and recirculate. And this turned out to be true not just for dendritic cells, which we hypothesized, but also for B cells and T cells. So the whole immune distribution was really massively altered from these sites. So we were wondering if this might be some crosstalk between immune cells and their neighboring stromal cells or even lymphatics, or if it was a general obstruction of flow. And so to test that, we went away from immune cells as the trafficking cargo and just used fitsidextran. And to do this, we worked with initially with Mike Davis to develop a system, and then carried out experiments in our own laboratory. And this work, all of the work that I'm showing you now, is carried out by Raphael Chepelevsky with the help of Bernd Senselmeyer, who is focusing on imaging in our group. And so what they developed was a system where the mesentery is pulled over a chamber And then Rafa used a specialized syringe to put 1 microliter of fitsidextrin into the Peyer's patch. This, as you will see in the movie, immediately the pressure of that injection leads to filling of the lymphatics of the intestine, and we would watch outflow of those collecting lymphatic vessels to the draining mesenteric lymph node. So in the control mouse, and I'll admit these experiments were done during COVID, so actually only one person could be in the room. So Rafa made the injection, then he went and turned on the video recording, and even in that period of time, we see some of the tracer has moved out to these lymphatics that are nearby the site of injection, as well as already starting to fill the mesentery going toward the lymph node. But the lymph node is initially empty, and then as transport occurs, in fact, in just a few minutes, the lymph node begins to fill up, as we expected. So that's the control littermate. But here's the unknown, the mouse that gets these tertiary lymphoid structures and has the Crohn's-like ileitis. And what he discovered were 2 important things. One was that, in fact, even cargo that is not cellular but molecular, like this fitsidextrin, is prevented from going to the lymph node. It never went to the lymph node. We would wait for up to an hour in multiple mice. And then also note that we start to see backflow and leakage. So our hypothesis was maybe there would be obstruction and leakage. Perhaps leakage could drive, fat expansion. That's something we're really interested in. And we observed this backflow. Now, backflow is a problem of potentially of altered pressure as well as altered valve function. Good valves should not allow backflow to occur. And so with Mike Davis, Mike actually used a micropressure recording to look at pressure just afferent to those tertiary lymphoid structures, and found that in the mouse with ileitis is very high. So, so pressure is elevated by the TLOs, and that sort of makes sense because, even in the images here, they actually look like pressure cuffs, in my opinion. This is very different than a lymph node would be. You can see that when a lymph node develops, during development, actually the lymphatics don't, the lymph node doesn't hang on to the lymphatic the way these TLOs are hanging on, but you actually see a very integrated structure. So the TLOs behave quite differently than lymph nodes. So the question then became, what's driving this change in, valves are no longer as functional as they should be, and they're also starting to sprout lymphangiogenic sprouts to facilitate TLO assembly. And so what we know, of course, is that valves are driven by a key gene expression that confers valve identity, is partly driven by oscillatory shear stress. So that oscillatory shear stress that occurs allows for FOXC2, for example, to be elevated. And we know that if we even just put, and here's some experiments that we did in collaboration with Josh Gallen and Yi Yang, if you just have lymphatic endothelial cells in culture and add flow compared with static conditions, FOXC2 will elevate, connexin 37 will be increased, as well as other genes like KLF2. And this essentially confers a lot of the properties of valves and valve identity. So what we did in these cultures is then add a concentration of TNF that we know to be physiologically relevant to ileitis. And what was very striking is that the presence of TNF in this oscillatory shear stress environment actually turned off, and some of these genes were just essentially completely turned off by the presence of TNF. So we don't completely understand how TNF interferes with this signaling, but there is a possibility from the literature that it may interface here, and certainly it appears as though TNF turns off valve associated genes driven by oscillatory shear stress. So TNF, a key mediator of ileitis, in fact, one of the major targets of therapies to treat Crohn's disease, can reprogram lymphatic endothelial cells to facilitate tertiary lymphoid organs to form. That, in turn, causes them to form right along the lymphatic, as depicted here in a schema that we drew from our findings. And that would drive lymph leakage, which we think might affect adipose tissue deposition and also alter the immune response. by altering this ability of cells to even get to the right place. But lots of questions remain, and really we still are, working on, and I'll just make a few remarks on this, how are TLOs associated with disease pathology. We want to dive deeper into these signals. And ask if valve identity, loss of that identity is sufficient to drive lymphangiogenesis, or what other signals might be needed. I do want to point out that so we're trying to grapple with the question particularly of what do these TLOs contribute to disease? Are they protective, or are they promoting disease progression? And I'll just throw out the idea that in fact they may in fact do both. A classical case of the two-edged sword, if you will, that we often use as a descriptor of inflammation. So I'll talk with about, and this is again under study, so at this point partially speculative, that the fact that we saw this leakage and backflow. So think about, backflow potentially spreading a disease entity that might be in the lymph from one region of the gut to another region of the gut, which otherwise maybe not have been inflamed. So in that scenario, the TLOs could drive disease progression. And so what is quite interesting from the clinical literature is that retrospective studies would say that if you remove the mesentery as compared with not removing the mesentery when you remove the intestine, so both of these surgeries have been in play when people are having a part of their intestine removed. Sometimes the mesentery is removed and sometimes it's not. So it looks like removing the mesentery actually with the surgery, so mesentery included with the removal, actually does appear to extend the time or prevent the need for a recurrent surgery. Many patients have to go back to surgery and they just keep having the problem in over, so it spreads. But maybe getting rid of the mesentery prevents the spread. And we wonder if that could be because of, for example, maybe backflow drives the spread from place to place. On the other hand, a hypothesis could be, to kind of harken back to my introduction, that TLOs actually make inflammation, constrain the dissemination of inflammation. So here you see the intestinal lymphatics in green. Adding these TLOs upstream of a lymph node might add extra firewall, if you will, extra filters that would prevent dissemination of inflammatory mediators. And we know Crohn's patients will have disseminated disease manifestations, like sometimes skin lesions, bone loss, muscle loss, etc. So one idea is that in fact these might be protective. So I'm just going to very briefly say that we're testing this question, and Emma Ehrlich is doing that by leaning into the literature that says B cells expressing a molecule called lymphotoxin alpha might be necessary for lymphoid. It's often necessary for lymphoid tissue formation. So she asked if in fact B cells expressing lymphotoxin alpha are necessary for these ileitis-associated tertiary lymphoid tissue to form, and the answer turns out to be yes. And she's currently writing up a manuscript to delineate the outcomes of the loss of TLOs. And I want to just make a couple of points without really, showing her data. You can look for that in the literature. But I've shown you that lymphotoxin alpha is required not only for lymph node development, now we're finding that it's required for these TLOs to form. It could be that the ligand is the lymphatic endothelial cell, so we're testing that question. But I've also talked about the role of TNF in the disease. And so I think you can immediately see when you look at, these are related molecules, and some lymphotoxin alpha can exist in two forms. So if B cells are making lymphotoxin alpha, this is the form that would drive lymphoid tissue development, but another form actually binds to the TNF receptors that are driving ileitis. And so we are very interested in the fact that the therapeutics that don't seem to work to treat ileitis, these are the major therapeutics that you see commercials on TV. Two of them are used to treat Crohn's. Those target only the TNF, but the one that targets lymphotoxin alpha is used for treating MS, and it's used for treating rheumatoid arthritis, but it doesn't work in inflammatory bowel disease. And we're curious if it may in fact be partly related to the fact that it also, by the fact that it's a TNF receptor 2 fusion protein, would not hit only TNF, but also lymphotoxin alpha, which we are finding is connected to the TLO formation. So I need to end. My last slide is just to say, tertiary lymphoid tissue is a really popular topic. Another area that I'm not going to touch on except for this one very quick slide is their role in combating tumors. And there we think of them as often being associated with tumor rejection. I think that's a super interesting area. What we're very curious about is, do they, in the case of tumors, would they do the same thing that we're seeing in ileitis, can change the tumor interaction with the lymph nodes? And there might be scenarios where that is beneficial. So there's really a lot of areas where there's activity and new tools to address TLOs and lymphatic connections. So let me just end by giving a shout out to the persons in the lab who carried out this work. I've particularly mentioned already Bernd Sinzelmeyer and Rafa Chepelevsky, and then more briefly Emma Ehrlich's work. NAVBO members who were critical to this work, shout out to Mike Davis, Ying Yang, and Josh Scallon. And Rafael Chepelevsky has been in the lab now and is finishing up. And I thought I had it here. I must have put it on a different slide. He is now starting his own laboratory. So the Chepelevsky laboratory opens July 1st at the Medical College of Georgia in Augusta, where Klaus Ley has started an institute for immunology. So, if you're interested in joining an up-and-coming lab, check out the Chepelevsky lab. So I probably went a little bit over, and I'm really, really sorry. So I don't know if we have time for questions or not. Thanks a lot.
Thank you, Gwen. That was a great talk. I think people are typing in their questions. There's we'll have quite a few for you, but maybe I'll have Sharon send them to you at the end, and you can answer people directly over email.
I will do that. Yeah, my apologies for going on.
No, you're fine. Thank you so much for a wonderful and engaging talk. Really beautiful work. So thank you again, Dr. Randolph. And so at this point, we're going to stop recording the webinar.